we will be talking about the translocation into the mitochondrial matrix. First off are some important things to note. Mitochondria have two membranes, an outer membrane and an inner membrane, both of which are composed of a phospholipid bilayer. According to the endosymbiosis theory, one would be the original bacterial cell membrane and the other would be the vesicle membrane that surrounded it when it was engulfed by the eukaryote. The signal sequence located on the protein, known as the N-terminus, is made of 20 to 25 amino acids. HSP70 is a chaperone protein that prevents the protein from folding before it goes through the translocon.
The use of HSP70 means that the process is post-translational. When we talk about TOM, we are talking about the translocase of the outer membrane, and TIM is the translocase of the inner membrane. An H+ gradient and the hydrolysis of ATP help get the protein into the mitochondria. MPP stands for the mitochondrial processing protease. Here is the outer membrane, the inner membrane space, the TOM complex, the TIM complex, and the matrix.
Proteins that are already made from a ribosome have HSP70 chaperones attached to keep the protein from folding. The hydrophobic portion on the signal sequence then has a weak attraction to TOM20. Then the protein has an attraction to TOM22, which strengthens the attraction to the complex and moves the protein closer to TOM40 due to the attraction. The HSP70 chaperone proteins are then removed. No one is certain how this is done. The TOM40 then allows the protein to enter the inner membrane space. The protein is then attracted to TIM23 and TIM50 of the inner membrane. These act similar to TOM20 and TOM22.
The protein is then pulled towards and attracted to TIM17 and TIM44. Then the protein moves into the mitochondrial matrix. As it enters the matrix, HSP70 chaperones attach and then pull the protein the rest of the way out. ATP then binds and changes to ADP. This removes the HSP70 chaperone protein. Finally, the MPP then cuts the signal sequence. The protein then folds to its appropriate conformation.